Okay, so let me first thank Sandeep and Devan for the invitation, and this has been a really wonderful conference because I got to learn many things which I really didn't know about. So what I'll be doing today is that I'll be talking about one small study we have done in relation, somewhat distant relation to LLPS, and then I'll be talking about something which we are recently doing is really trying to understand simple IDPs, that how we can think about the landscape of IDPs from a simple theoretical background or theoretical point of view. But let me first start with the what I said. The first thing first, in IACS we work on many different problem, which is mostly, I would say, it's not a biological problem, it's basically a physical chemistry problem. That means we look for problems in biology which can be solved by understanding of physical chemistry or physics per se, and then we try to find out some of the underlying principle what is working there. Mostly that is the theme, and these are the different projects we work on right now. Okay, so first thing I'll talk about something we have done in relation to LLPS is that little bit introduction. This you all know by now. So these are very important from different perspectives, but of course regulation is important. If you uh, get into dysregulated thing, then you get into trouble, having disease and stuff. Like, people have done a lot of studies to show that there are weak interactions which are playing the game here, but you need to understand that probably in all the LLPs, all of these are playing the game, but then what is the weightage of one another, that is actually dictating what you are going to see and what you are not going to see. Okay, so some of the experiment is one is really interesting. It's called reentrant behavior. That means if you have, let's say, protein RNA kind of condensate, for some of the systems people have seen, if you start with very low salt concentration, you really do see those aggregates or those LLPS behavior. Then you go to intermediate salt concentration, nothing, it vanishes. Again you go to higher salt concentration, you again tend to see those. This is a very interesting reentrant. So it has something called I mean, in temperature language is LCST, and lower critical and higher critical temperature, something like that, in terms of ionic strength and stuff like that. Interestingly, in one of the recent paper, they have designed a very simple peptide, charged peptide. It is proline arginine repeatedly for 25 mar, and they showed that this can actually self aggregate to give you those kind of thing you see in LLPS at 2.7 molar of very high salt concentration of KCl or NaCl. So what in this study we are doing is very simple. What we are trying to see, we said, okay, we'll try to decouple some of the interaction going on in this kind of studies, and possibly we'll be making some predictions to understand what's really going on here, why you are seeing these kind of things. First thing, we'll be thinking about only interaction at the dimer level. Okay, we'll try to form dimer, nothing else. At the salt concentration which is closer to them, and our design is very simple. It is Rx, Rx. R means arginine, X means it can go from glycine, alanine, proline, or phenylalanine. We'll also prepare other system where it will be EXX, E means here glutamate, okay, and then X can also vary. Okay, these are the type of system we are thinking about, and we are doing self-dimerization, nothing else. How do we do that? Computationally, you can of course do that using something called metadynamics. It's a free energy simulations that we try to find out using our order parameter what is the free energy of the system. We have taken order parameter as the number of contacts the two dimer can form, and that we have biased in our system. We are varying this from 10 number of contact 10 to 100 and 10, and these are the some things we use some kind of cutoff to show that there will be some distance criteria where there will be a contact, and out of that, if it goes beyond certain distance, there will be no contact. Okay, so let's see what we find. First thing we found is that when your system has arginine, if you have glycine, these are two temperatures you are seeing, they don't show any propensity to dimerize at all. Only when you are increasing the level of hydrophobicity, That means going from alanine to proline. Remember, arginine is constant. It's a repeating things, arginine, proline. You find at high temperature, actually you do see that higher contact forming stuff is getting stabilized slowly. Okay, but what is interesting, you don't see the same thing when you have glutamate sitting there. That means if you have glutamate, if these systems are not showing that propensity. All this is calculated at 2.8 molar NaCl solution. We have shown for arginine alanine system, we have just for purpose, we checked what happened for 1.4 molar. We have seen that propensity is actually decreasing. That means you do not find that much aggregation propensity what you have find for 2.8. But the message is that for arginine system, you do find a propensity for dimerization, but not for the glutamic acid system. We said, okay, let's see that what are the things we can calculate? Calculate many things. Long story short, nothing is so different between the two systems. Number of ions, Rg, number of water, also not very difficult. But what is really important here is that if you look at the residence time of water, you find there is a real difference. Around arginine, water doesn't like to stay too long, but around glutamate, they do like to stay really longer. I mean, literally longer, like 2 times or so. And that is actually reflected. These are, don't go to the numbers and plots here. You see the energy values, these are very pretty small, 1 to 1 point minus 3, 3.3, but only interactions which is really important is actually glutamate and water interaction. That is actually strong interactions. No other interaction in terms of ion or water with all the side chains are important. I mean, important means number wise. It is the glutamate which actually prefer to interact more with water. And that is probably the reason it doesn't want to go to a state where it want to dimerize or go to a state where it can form aggregate. Okay. Is there anything else we can think about? Yes. You can see that these are actually side chain interactions. If you see side chain, these are for arginine system. You see more or less they are very similar at 2 temperatures. Only for proline at higher temperature you are finding little bit more interaction edge. That is probably because they are more hydrophobic interaction coming into picture and making the interaction more stable. But what is important is that if I want to now calculate self, like how arginine and arginine is interacting or how glutamate and glutamate is interacting, what you find, arginine, there is a favorable interactions when they are collapsed. But for glutamate, you don't find such interactions. So it is a mixed effect. It is a solvation, water which is preventing not to go there. And other thing is that because arginine has this pi character, probably it has certain amount of indirect hydrophobicity coming into the picture which people have talked about earlier. That is making the system with arginine more prone to dimerization, not the glutamate one. We went ahead and did something else also. For phenylalanine system, arginine, phenyl, glutamate, phenyl, we just did a simulation putting some of the 5 or 6 chains and tries to see what happened. We have seen the similar thing for arginine system. We found they are all coming together and forming a single droplet kind of stuff, but not for again the glutamate system. And we characterize what is playing here. Of course, here cation pi is playing much more important interactions than any other, because you can see this arginine and this phenylalanine from 2 different chains, they can form these cation pi interactions, and solvation is also probably here hindering that effect not to come into the picture because cation pi is not there. This is what I wanted to say for this very simple system, that how we can understand what is going on. Okay, and probably what I prediction was, I am telling that if once make us things with glutamate, probably that is not going to go for a NLPS kind of transition, but we don't know, of course. This is kind of a signature coming from our work which we have done in 2023. Okay, now again, now forget about NLPS little bit, because this is something different I am trying to talk about. It is about protein folding again. So we know protein folding what it is. Amino acids, they can form chains, it can go to 3-dimensional structures, all this stuff we know. I will not bore you with this, you all know it very well. And you also know Anfinsen's experiment, famous experiment, that you denature the protein and fold it back, it always goes back to the same structure with this particular protein. And then it says that information contained in the protein sequence is sufficient to determine the protein structure. And thermodynamic hypothesis is also there. It says the native structure is the global minimum is the free energy surface. It talks about two things. One, sequence, if you know, that will give you a unique structure and thus function. And second is that this means it is not only telling that it will be in the global minima, it is telling probably the folding pathways are also defined, because it does not necessarily only define the minimum of the free energy potential, but the whole landscape from going from the unfolded state to the folded state. Okay. Now, the question is that this problem then has two parts. One is structure prediction. That means if I know the sequence, can I tell you what is the structure? That part, people have done a lot of work. Let's first acknowledge that physics-based model has done fantastic job and everything. But then fortunately or unfortunately, I don't know how to say that, but in 2024, you know that Nobel Prize has gone to the, AlphaFold, Google DeepMind AlphaFold, solving this problem using AI. But don't go with their all the saying. This model only works when you use multiple sequence alignment and pair correlation kind of stuff very heavily, otherwise it doesn't work. So it is not necessarily a full black box. Okay, physics is already there. Okay, first thing first. So there is structure prediction part is probably fine. What about the other parts? It is the how really the protein folds. So for that there is Leventhal paradox. We all know protein can astronomically high number of conformation possible. How it will select one? It will take too much of time. But then protein actually folds within biologically relevant time, which is pretty small. And this old work by Professor Joanjic, Atula Jabo and Biman Bakchi, they actually talked about this, and they said really interestingly that if you just think about a small bias, that means all these amino acids can have many different interactions. You just think that if they are in native state, different configuration one amino acid can have. In native state, this is 1 kT bias or 2 kT bias compared to all the non-native states it can exist. That solves the puzzle. That says if you have that biased surge, that time scale actually can come down to as small as 1 second. Okay, so it is, that means it is saying it is not a random search, it is possibly a biased search. How it is biased? They thought it is because of the native state which is being native interactions or native configuration is being stabilized compared to the all non-native states. Great. Other physics-based model of understanding protein folding, one is Ising model. They are doing a binary kind of stuff. That means they are taking protein as folded or unfolded. But more sophisticated model is called spin glass model, where you can have both ferromagnetic and antiferromagnetic coupling within the spin system, and that gives you all kind of frustration you can have in your system. And you know the spin glass model can give you multiple local minimas and stuff like that. With some interesting variation of that model, this Brinjelson and Walliness is a famous paper of protein folding spin glass. They could show that if they think all the native state have some, all the native interaction have some slight stability than all the non-native interactions, where non-native interaction can be considered as a random energy with some Gaussian distribution and having some minima, having some average and distribution. I will come back to that. Then you can show that system can have phase transition from unfolded to the folded state, and there is something called frozen folded. I will come back, but don't worry about it. Folded and unfolded. Okay. But you understand something, that paradigm of classical structure function, sequence structure function has changed. Because now we have metamorphic proteins. We know the same sequence can give you 2 structures or maybe more structures, giving you more functions. If that was not enough, then you know that there are proteins which doesn't have any structure. So that means they are kind of IDPs. Okay. So things are now loose in terms of that particular Anfinsen's way of talking, that is becoming pretty loose. It can have, let's say, infinite number of structures. I don't know how many structures, but sometimes, not always, they can be fuzzy structure also. Sometimes they can actually bind to a target protein and give you a real three-dimensional structure while functioning. All those IDPs I am talking about, generally they do not have any structure, but with target they can go to a particular structure. It has been shown. Idea is, can I get a theory which can explain everything at once? Okay, this is just a try. We have done it for system where we said we have string of n amino acids. Each amino acid can have n number of native states, multiple, n can be vary, and 10 non-native states. That is fixed. That means I have some 10 non-native states, but it can have multiple native configurations, not one. Okay, and that is my n because I want to do it general. Okay, but I will present first n equal to 2. Okay, because that is more tractable. So idea is very simple. I have string of n amino acids. Each amino acid can have 2 native interactions and 10 non-native interactions. That's the simple thing. If you think that way, okay, I'll just go through. Don't go to the equations. I have put it, but then I'll just go through what I'm trying to say in a simpler terms. So what I said, 10 you can have nu which is equal to 10 our non-native configurations apart from n native configurations, and we are doing for n equal to 2 state, n equal to 2. That means it can have 2, let's say, for the time being. Now energy terms we have defined in simple. A particular amino acid at a particular position in which state? Is it in native state or is it in non-native interactions? And then Ising pairwise term. Is the two residue one after other, they are in native or one native, one non-native, or two non-native, any kind is possible, right? That interactions terms are defined by some J term. But protein is three-dimensional, so there can be tertiary interactions also. So it will also depend whether two tertiary interactions which are not close in the structure, I mean sequence, but close in the three-dimensional structure, whether they are native in nature or non-native in nature, depending on that there is an interaction Kij I can define. This is very simple thing. So then any configuration of protein can be written like this, very simple. It's the term which is residue level, this is pairwise, and this is all three-dimensional structural level. Yes, that's the model, very simple model. Now what I am asking, that let's say I have n0 number of states, n0 number of residue is in the native state, while my length is n, some fraction is in the native state. That can give you many different kind of configurations, right? Energy, having different energies, they are generally not correlated. If I think about random energy approximation, so it will be kind of a multiplication of all probability of having that energy of different energies, and I can multiply that. Now question is that this I can now, using the spin glass way of doing, Derrida and others have done, we said I will go to a stochastic approach. That means I say what this probability should look like. Because it is no correlation between one and other, generally it will be Gaussian with some mean and some standard deviations. Okay. Fortunately, if we think something interesting here, then we can say what it is. Now, how I am doing it. So this is my mean and this will be my standard deviation. How I am doing it? Very simple. I said if they are in the native state, they have this certain energy, either epsilon 0 or J or K, depending on what kind of interaction I am looking at. If not, they are drawn from a distribution whose minima is suddenly little bit higher in energy with some distribution delta E. Okay, you can do a simple probability studies that if I have n0 number of states, how possible that next one will be my native or non-native. That way you can just do all these calculations to get very simple expression for this average of the distribution and the standard deviation of this distribution. Yeah. Which one? They are independent, yes, yes.
equilibrium, I can just write down the Boltzmann rate, right?
Yeah, equilibrium you can write down Boltzmann, but where I am going is that I am trying to find out what should be the distribution, and from there I will actually talk about something called microcanonical ensemble, like density of states approximation I will go. From there I will calculate the density of states from the probability distribution. And
so your ensemble is because I thought somehow it's a canonical. So
no, it is microcanonical.
You are considering a micro.
Yes. Okay. So now, once you have that, you can do that. Okay, you can write down. Just take it for now. From there, what you can define something called density of state. How many states are there at a particular energy, and that will have certain expression of this kind. Okay, forget about this for the time being. If you have this, if you have density of states, statistical mechanics tells me if I take log and then I maximize that, that will be my entropy. As simple as that, because coming from microcanonical ensemble. So what I can say that from here, what I can define is actually the entropy of the system in terms of the order parameter, which is simple is n by n0, n0 by n, or for a particular relative state it will n1 by n, right? Because you see, n rho is the total fraction of native fraction, and rho 1 is a particular one which is state 1. So 1 minus that will be simply your rho 2, the other state, how much will be the fraction. But now that is the beauty. Once I have that, I can use this expression to get back to the canonical ensemble and define my energy entropy and free energy of the system. Once I have that, I can define the derivative of those. Once I have those derivatives at different value of L, that means this interaction differences, you see that how much is the native state is stable compared to the non-native state, I can have a phase diagram simply because I can solve this simultaneously. Yes.
Saying that because it's microcanonical.
Yes, it has to land. Yes. Yes, exactly. Nothing else. So, but you understand that, I mean, anyway, those, I mean, at least statistical mechanics says that even if you go to canonical ensemble, it is a delta function around that average value anyway, right? There is equivalence of different ensemble. Okay. So now, if you have this, you can solve that, and at different condition, you can see the phase diagram, what should be your rho and rho 1. And that's what we'll do. But before that, just for the clarity, we actually derived for n. Any n possible you can think, and we define all the derivatives and everything, and then you put n equal to 1, n equal to 2, n equal to 3, n equal to 4, 5, and get all the equations you want and solve for it simultaneously and get the phase diagram. We use simulated annealing, okay, for getting this all the phase diagram and everything. That means the maximization problem. Okay, now doesn't matter what I said earlier, just the result. What it's showing is that this is L minus L bar by T, means it is the tertiary secondary interaction taken together, L minus L bar. It is telling how much my native state energy between two residues, that means pairwise, how much it is stabilized compared to my non-native interactions. And this says how much is my width, because that is the entropy. So how much is my width? It is the average value wise, how much is the difference? And for non-native, there will be distribution because it is random. How much is the width? That is the two parameter I am varying here. If you vary, what you find really interesting? System initially in this region will be in the unfolded state. Then it will cross a phase transition, first order phase transition, and it will go to folded region. How you can check that? You see rho low fraction of native contact. At the onset, it jumps to 0.8 or 0.9. Okay, rho is total number of fraction native contacts. So two things I am finding. One, all the systems, this is for 1, 2, or 5, they are actually crossing this phase transition. In terms of total fraction of native contact, they are all doing this phase transition. But what is interesting, you see, as I increase my n, I am shrinking my folded region, right? Now think about, if your n is really high, what is going to happen? You will shrink really too much, then at a physiological temperature, which will be somewhere here, even if you have 10 number of native states possible, you will be in the unfolded state as far as the phase diagram is concerned. And that is your IDP. Okay, that is your IDP. But when n is limited, then what happens? Like n 1, 2 or 3, you'll see what happens. Look at the folded configuration for n equal to 1, 2, 3, 4, 5. Folded configuration means I am in the folded state. I am checking the health checkup of folded state. How? I know for total fraction they have all crossed the threshold. It's like folded. But I know for two state, there are two state possible. So I have to check each of the row. So the highest row value, I said row 1, next one I said row 2. Okay? You see, for 1, 2 and 3, 1 is of course singles basin. It's a two basin, two possible state, three possible state. All of them goes to a high value of row 1. That means when they fold one of the particular structure, one of the three. However, when 4 and 5, first of all, folding region is shrinked. Possibly they will not fold in normal region. Even if they fold, none of the structure will predominate. It will be a mixture of states. How do you see that? The highest row is decreasing. Here, the second lowest, second highest. What happens? Once it is folded, for row 2 and 3, there is a demixing. That means row 2 is decreasing, row 1 is increasing, and for 3 and 4 and 5, it is a mixing. That means it is the other roots are also increasing. That means there is a mixture of states in the folded state, if at all it goes to folded state. Yes.
I'm going to try and paraphrase you. Please say whether what I'm saying is right.
I think I am not doing good with time. Yes. Yeah.
Maybe I'll do it later.
No, just go ahead, please.
So, for every residue position, there are multiple conformations possible, and they are all at equilibrium.
Yes.
And then for the next one along, there are again multiple. The number may be different, but they are at equilibrium.
Yes, you are right.
And so on, right? And the IDP has not complete structural freedom,
Yes.
but a limited structural freedom where
Right.
at each position along the chain, you have any one of 1, 2, 3, 4,
Yes,
5,
yes. That's my point. Is
right? And then when it all sort of precipitates into a single structure upon binding to the designated cellular partner,
I'll show that.
does it fall like one?
Yes,
does
yes.
cooperativity?
I'll show you. I'll show you. That is the next thing. So this is fine. And all these plots are the same. Don't. I will not waste time. But what it is showing, at least in my setup, up to n equal to 3, it is all like singly folded. N equal to 2, what will happen? At free energy, we'll have two basins. One fully one state, another fully two state. N equal to 3, we'll have three basins. But each of the basin has one state. Right? Mixing happens when n is increasing beyond 3. Okay. So now, you see, this is just a comparison of 1 and 2. I will not waste time. But look at this. For n equal to 2, I can get actually the free energy diagram. I have, because for n equal to 3, it will be complicated. N equal to 2, you see, in the unfolded region, you do see unfolded state is the stable, but then you have two basins which is unstable, equally stable two symmetric basin. Yeah, 3 is fine for me. At transition state, where you see the spike, you see all the three basins has very similar like stability. But in the folded state, you find two symmetrically stable basin folded. It's like metamorphic protein. Same will be for n equal to 3. Okay. Now this is interesting. What we said, for n equal to 4 model we have taken. Okay. So this red line you are seeing already shrink to 1 folded. This is unpart model. I just plot the n equal to 4 first order festration. Then we should just do this simple thing. Out of the 4, designate one, the first one, and say there is a bias of 0.4 kBt or 0.5 kBt or 0.6 kBt. To say those bias in the native structure will come when it will bind to a partner. Nothing else. Just put a bias for one particular state out of the 4. Very little bias. Okay. That is only in the residual level, not in the pair level or anything. We have not done that. I mean, if we do pair level, it will be complicated. Okay.
Complicated for one more reason. In alpha-helical structures,
Yes, you are right.
use
Yes. Yeah. I get your point.
I2 I plus 4, but in leadership
Yeah,
they don't.
you are right. So when I do that, what I find that you actually regain folded state. When you put that bias, you are actually going towards your folded state. Now folded line goes backward. That means you are regaining the folded state. That means there is a propensity that if the position of the sequence is here temperature wise, earlier it was unfolded region. Once you put this biasing, it becomes folded. Good. But how it is getting folded? Folding is exactly same as one state folder. That means you find only the state you biased that is becoming folded, all other demixed. That means what I am trying to say within this whole thing, that if I have a n-native state description, there I can actually show that when n is finite but not one, more than one, there I can show that system can still exist in a demixing limit. That means it can go to metamorphic kind of states. But when your n increases slowly, your phase transition line is shrinking, making folding propensity closer to 0. That means at a physiological temperature, your unfolding probability is more, unfolded state probability. That means it will be unstructured. But moment you put bias on one state, phase diagram also shifts towards the right directions and you get folded to the actual folded state. That means which one you have biased. Okay. So we are trying to say that using this, we are kind of giving you a theory where you can actually take care of everything. Okay. Now, whether it is fine or not, that's up to judgment later on, but this is what I wanted to talk about. But I think I should talk about the first part. LLPS Devashish Saha, he has done this, and the second part is Rohan Moitra. He is currently working in my group, of course, doing on other stuff, and DST ACRB for funding, ISCS for some amount of computing, and thank you.
Thanks. Thanks for a wonderful talk.
Hi, very nice talk. So I was wondering for the re-entrant behavior. In the paper, I have seen that when increasing salt concentration, in the low concentration, the electrostatic interaction is the main driver. And when increasing the salt concentration, now the electrostatic interactions are hidden and mostly the residual hydrophobic interactions play a role in the high salt phase.
It says interesting line. I have seen some paper. It says hydrophobic and non-electrostatic. So that means they really do not know what is happening. It's something like, I would say hydrophobic and indirect interactions through ions and water rearrangement. That is what is happening probably. Okay, anyway, yeah, go ahead with it.
So I was wondering, have you looked into extremely hydrophobic amino acid residue and
We have looked for phenylalanine. There we found cation 5 is
cation
the
5.
real factor, but we have shown for glycine, alanine and proline, and we show that for proline actually the experiment is also on proline. It is showing the more propensity. See, the point is that arginine is making sure they come together. Proline is making sure if you increase the temperature, hydrophobicity takes into account. Because
if there is valine isoleucine.
Yeah, it will be there. I mean, I cannot check all that.
Right. I was like thinking in terms of more hydrophobic.
Yeah. I mean, I'm sure that those will be there effects, but we need to check. Okay. We cannot just say that it will be there.
Thank
you. Talking about the Leventhal paradox, most people have paid attention to how within finite time, through some pathway, an accessible local energy minimum is attained, not necessarily demonstrably the global energy minimum.
Global energy, yes.
And all of it actually started with Anfinsen's demonstration of refolding. But there's also a very large part of the community which asks a very inconvenient question: where is the scope for refolding to occur in nature? So, an alternative explanation, right, which addresses the Leventhal paradox, is co-translational folding on the ribosome.
On the ribosome, yes.
As the chain emerges from the ribosome, progressively longer stretches of chain developing through vectorial addition of amino acids during translation is actually creating at every stage of addition of amino acid a new conformation energy
Yes.
landscape.
And then that can trace back.
And if that goes to the thermodynamic minimum at that stage,
It will stay there.
from there, it may be a short hop to what happens when the
Yes.
next amino acid is added, and the next, and the next, and so on. So
Yes.
therefore, what refolding, when it occurs, and it doesn't occur, I study a bunch of proteins from hyperthermophiles which do not refold, right? So when it occurs with mesophile proteins and with single domain proteins,
Basically for small globular.
small globular proteins, single domain proteins,
Yes.
no disulfides, no prolines,
Sure,
and so on.
100%.
Right? So where it occurs, it probably recapitulates co-translational folding.
Transistor. You're right, you're right. Yes.
That means nature has designed it for folding from the N-terminus to the C-terminus through gradual production. And when the whole chain is there and refolding and doesn't know what to do, the N-terminus starts off
Yes.
doing something autonomously.
Autonomously.
And progressively, as you go towards the C-terminus, they pack against those preformed structures.
And
And so
it is also known that when you do co-transitional folding, it actually can sometimes go to something very different structure,
yes,
which is not the native structure
Yes,
actually.
that's
And
right.
then
in fact, for cetylysin and for alpha-lytic protease, it has been shown
yeah, I know.
that if you refold it, it goes to a totally different structure.
Yeah.
So therefore, when an IDP
Mhm.
forms a native structure after binding to its designated cellular partner,
Yes.
there is a possibility that the precipitation, I'm using the word precipitation here in
I got it.
terms of English, not in terms of science, right? When a state precipitates for each one of those
Yes.
residues, it might actually also recapitulate co-translation
Co-transition. Oh, you are right.
from one end of the chain.
Yes, yes.
So that sudden shooting up
Mhm.
may be happening residue by
Residue
residue
by
residue by residue.
Yeah, we haven't done the dynamics on the landscape yet. This is just the free energy part. We can do the dynamics on this. We have a plan to do it, but that is much more difficult, by the way. But we have a plan to do that. This will be complicated, but we'll do that. The dynamics on this multi-dimensional landscape, how it works.